All right, thanks everyone for being here. It is my great pleasure and honor to reinstitute our speaker seminar series. It's a series that we started many years ago, but then kind of fell to the wayside during COVID. So this is our first guest speaker that we've had since COVID. And he is going to be speaking to us today about brain drainage and microglia in neurodegenerative disease, which has become very relevant for us here at the Foundation as we talk about how we can use focused ultrasound to treat neurodegenerative diseases. So his name is John Lukens. It's my great pleasure to introduce you to him. He is an associate professor of neuroscience at the University of Virginia. And we are also here to celebrate his recent appointment as the director of the Harrison Family Center for Alzheimer's and Neurodegenerative Research at the University of Virginia as well. So congratulations, Dr. Lukens. And he is also the husband of Jessica Lukens, who works here in development. So we're really happy to have John here today to talk to us about this really cool and relevant topic for us. So thank you, John. I'll let you take it from here.
All right, cool. Thanks, everybody, for coming out today and listening. I was just going to probably share a couple of things that are ongoing in our lab and also introduce you to some emerging concepts in the field of neuroimmunology, and mostly be talking about traumatic brain injury and Alzheimer's disease, but doing a little bit of introduction about what it means in terms of neuroimmunology to provide some kind of concepts to give you some background. And I know that cancer immunotherapy is also a big mission here at the Focused Ultrasound Foundation. So I thought there's certain themes there that are very relevant and pertinent to cancer immunology too. So that's how I was going to go about it. You can stop me at any point. It can be really laid back and informal if you want. I'm not quite sure the background in the whole audience. So if I go a little too deep with the jargon, please stop me or ask for clarification. Okay, cool. So there's been, is everything okay? There's people coming on in and off. Okay. Okay, so there's been a reemergence in interest in terms of the idea of the immune system in the brain, and for decades it was thought that any immune response in the brain was a bad thing, and there was this idea that there was immune privilege, meaning that the immune system was cut off from the brain and that there wasn't very much interaction. And there's quite a few reasons that came to be. And I was just going to kind of go through a couple of these. But conceptually, it makes sense in the sense that the brain is full with postmitotic cells. So these are postmitotic neurons that hold our memories and they control all of our functions. But unlike, say, hepatocytes or your skin cells that undergo damage and that are replenished after they die, neurons, for the most part, don't come back. There is a little bit of adult neurogenesis, but it's not sufficient to keep up with neurodegenerative diseases. So knowing that neurons aren't easily replaced led to the concept that you want to prevent any kind of damage, collateral damage in the brain, to help sustain these very important cells that are absolutely essential to everything we do in life. I'm not going to talk too much about the various different types of glia, but in addition to neurons in our brain, which are hardwired to conduct basically information from one cell to another and control everything from breathing to cognition, we also have these other supporting cells. They're called glia. And they have, in the Latin, this means glue. Initially, it was thought that these were just cells in our brain to kind of hold everything together and keep the neurons together. Over the last couple years, probably 20 to 30 years, we've come to realize that these glial cells, which are a couple different types, we'll talk about the microglia today, have a very important role in terms of sustaining homeostasis, so health of the brain, and making sure those neurons can function. In addition to neurons being post-mitotic, glia are also quite long-lived. So any kind of changes that happen to them are things that could impact the brain for the life of the person. So this kind of led to also kind of lend itself to this idea that maybe we don't want to have inflammation or these immune culprits that could cause damage or dysfunction of those cells in the brain. I'm going to be talking about this in a little bit, but in a couple slides. But forever, at least to 10 years ago, it was thought that the brain didn't have lymphatics. So what lymphatics are these brain drainage structures, and almost every organ in our body has them. And this is a way for your brain, that organ to communicate with your immune system. So basically, they're basically pipes that leave that organ and go to where your immune cells are, and they carry antigens and waste, and that the immune cells help to are always constantly surveilling those. And it wasn't until about 10 years ago, it was actually at UVA by Yoni Kipnis's lab, which was down the hall from me, that they identified that we were just missing them in the brain. So most organs, they kind of go through the soft tissue, and they kind of right up next to them. Is that okay? Yep. Okay. But in the brain, we were missing them, and I guess you could blame this on the neurosurgeons in some ways, in the sense that when you do pull off the skull of the brain, the meninges are this very thin layer that often sticks to the skull, and this is where the meninges are, and this is where those lymphatics are, and basically we were missing them because we were basically, as we were cutting into the brain, we were just pulling off the lymphatics with the skull. And that was a major reason why it took so long to discover them. Most other organs outside the brain have these cells called antigen-presenting cells that are very important in terms of initiating and starting the immune response. Most organs have multiple professional antigen-presenting cells that can do this. The brain only has one, and it's called the microglia. And I'll talk a little bit more about some of our data implicating them as important players in Alzheimer's disease later. But these are, as I mentioned before, very long-lived, and they come from a unique progenitor pool. So they're hard to replace and manipulate, unlike other macrophages, say, in your lungs or in your skin. As I mentioned before, they're poor at promoting antigen presentation, which is basically communicating with other arms of the immune system to start an immune response. And where this becomes a problem is in terms of tumors of the brain. So since they're so poor at communicating and telling those T cells that you need to attack this cancer cell, or if it was actually an infected cell, you could also look at it in terms of infections of the brain. It really limits the ability to mount an immune response in the brain. Another major component that we've known about for a long time is that there's a very tight fortress around the brain that limits cells from the periphery getting in, cells like immune cells. And this is the blood-brain barrier. And this regulation of this endothelial, this entry port into the brain, is much, much, much tighter than most other organs in your body. So it's a lot easier for an immune cell outside of the lung to get into the lung or the liver, you pick your choice. And then there's also quite a bit of immunosuppressive cytokines. So these are factors that are continuously secreted that are basically damping down the immune response locally within the brain. So that's a little bit of, I guess, if you're thinking about, this has important implications both in terms of cancer immunology, neurodegenerative disease, pretty much any neurological ailment that one could face, and that focused ultrasound could be kind of piggybacked on to help. And this is a major reason why things like focused ultrasound in terms of opening the blood-brain barrier to help get those immune cells in, or drugs. So this BBB tightness is the issue there. And then also with cancer immunotherapy, people are using focused ultrasound to make the tumor more pro-inflammatory, since it's so hard to break this innate tolerance of the brain. Are there any questions with that? Or okay. Okay, so I'm going to be talking a lot about some of our Alzheimer's disease work, but just to give you a little bit of an introduction, 1 in 9 people over the age of 65 are diagnosed with Alzheimer's disease. With an aging population, these numbers are expected to go up. Obviously, as you probably all are well aware of, this is associated with the loss of cognitive function. And also the loss of those neurons, those cells that are important in terms of carrying memories in the brain. So the major hallmarks of AAD that are used to diagnose it are amyloid beta. I'm trying to get by. Okay, so amyloid beta, tau tangles, and these are neurotoxic compounds. So they basically cause the immune system to become activated to produce cytokines that could kill surrounding neurons, or they can also interact and eliminate the ability of the neurons to make connections. So eliminating these neurotoxic materials by various methods is a very attractive means to treat neurodegeneration and Alzheimer's disease in this context. And then the third major hallmark of Alzheimer's disease is something called microgliosis. And this is just a terminology used to indicate that the immune cells of the brain, these microglia, become hyperactive and they change morphology and they start upregulating a lot of different factors that could negatively impact brain health. So I mentioned all of this. This is the kinetics of the disease. So early on, you get deposition of amyloid beta. So this neurotoxic compound that builds these plaques in the brain. Soon after that, you see activation of the microglia. They're responding to the amyloid beta. And then decades later, you have the second neurotoxic culprit depositing in the brain, known as tau. And it's really at that point that the tau pathology starts, that you have loss of the neurons and you start seeing the early signs of memory dysfunction. So there are lots of currently pursued therapeutics that are trying to eliminate things like amyloid beta and tau from the brain, and also modulate or find the beneficial functions of this immune response to these neurotoxic. So that's in terms of the space of where Alzheimer's disease translational research is, it's kind of in this area. And where focused ultrasound's been coming in quite prominently lately is opening the BBB, so some of these compounds that are good at sequestering away these neurotoxic compounds can actually get into the brain. So coupling antibodies to amyloid beta and tau, opening the blood-brain barrier in regions where there's that neurotoxic material is kind of one strategy that a lot of people are doing and was featured on 60 Minutes a couple months ago. Okay, so where are we at in terms of studying this? So we're pursuing this in a couple different ways, and I've kind of already introduced this concept with you, but in terms of the diseased brain and the healthy brain, what happens is over time, you get this deposition of these neurotoxic compounds, things like amyloid beta and tau, that cause inflammation, overactivation of the immune system, and this ultimately results in loss of those neurons and dysfunction. And we're trying to think of ways of how to revert that brain that's full of these neurotoxic compounds that trigger their immune response back to a more healthy state. So the solution to this that we're pursuing and others is trying to find ways to improve the disposal of these neurotoxic materials in the brain. So one thing that we're working on and others is understanding how we can leverage this brain drainage pathway that sits above the brain to help pull things like amyloid beta and tau out of the brain to regain healthy tissue. And then the other area that we're trying to tackle this in is trying to promote the beneficial aspects of those cells that are going around the brain to chew up and engulf and remove that neurotoxic material. How do we boost the function of those? So those are the microglia. In a lot of ways, they're the garbage men of the brain. They help to both eat and kind of corral the neurotoxic material. So where our lab and others, and also a lot of biotech industries are trying to find ways and markers on these microglia cells to rejuvenate their function and to improve their ability to clear these things. And I guess an important concept to kind of talk about right now is, obviously, I'm going to be telling you more about our Alzheimer's disease research, but many of these concepts are also present in most other neurodegenerative diseases. So if you look at Parkinson's disease, for instance, you get deposition of a different neurotoxic material, alpha-synuclein, that has a very similar effect in causing inflammation and then damage to the neurons. So this idea of improving clearance and kind of finding that happy place where you get a beneficial response from your microglia is something that people are pursuing in the context of AD, Parkinson's disease, ALS, Huntington's, pretty much every neurological disease that you can think about. And I guess when you're thinking about some of these concepts too, there's also the other side of it. Like in AD, we're actually trying to turn down a lot of these pro-inflammatory things. But for brain tumors, there's, more and more emphasis is being put on rejuvenating or reinvigorating those exhausted immune responses in the brain. So you can kind of think about it as a balancing act that needs to be kind of fine-tuned depending on the disease that you're studying or that you're trying to treat. Okay, so now I'm going to tell you a little bit about some of the work that we're doing in terms of this brain drainage pathway known as the meningeal lymphatics. As I mentioned before, up to only 10 years ago, we didn't think that the brain actually had their own lymphatics. And then there's been a kind of an onslaught of papers recently that have identified them in this area known as the meninges, which is above the brain parenchyma and under the skull. And this is the tract that they run. They run right over the brain, and they drain to an area known as the lymph nodes. And that's where all the immune cells are. That's where your T cells that would help fight cancer and viruses are located. So since then, we've come to know that these lymphatic drainage pathways are very critical in terms of removing things like amyloid beta and tau, these neurotoxic materials, from the brain. So if you were to basically remove the lymphatics using surgical or ablation techniques, this would result in an increase in amyloid beta and also tau. And we also know that this pathway, as it is the case in other organs, is the major mechanism by which that brain organ communicates with the immune cells in the periphery, and that it can help to make sure that there's mobilization of other cells into that organ. So this is a project we started about 8 years ago that we're still working on in some capacity, and I'll kind of share some of the new directions that we're at with it. But this work was all started by a really talented ND PhD trainee at UVA, Ashley Bolt. She's actually still at UVA. I think she's a resident in internal medicine, but somebody that we're hoping to bring back on in our new center in a faculty role. But when we first started this project, we knew that the lymphatics were there. We didn't really know what role they played in the context of any disease. So this is rewinding back 8 years ago, 2 years after they were actually identified and their location, but we didn't really know what they were doing. So the question that she asked was, how does traumatic brain injury, so TBI, impact lymphatics? And if so, how does this affect disease pathogenesis? So how the brain responds to that injury. So we're using a mild closed skull model of traumatic brain injury. So when we hit the mice, we picked an area far away from the actual lymphatic vessels. We also use a very mild impact in terms of there's no cognitive issues. There aren't any issues in terms of motor coordination. It's similar to a very mild head injury that you might receive in a sporting event or from a minor car accident. And the first question we wanted to ask was how sensitive is this drainage pathway to traumatic injury of the brain? So basically, what we did was we hit the mice at a certain time point, and then we injected in these beads, these fluorescent beads that you could use as tracer to measure the functional ability of those lymphatics to drain. And then we injected those beads in every couple hours or days post traumatic brain injury induction to ask the basic question, how severe is the lymphatic compromise post-injury? And then also, how long does it take to return to normal functioning? And so basically what you do is you inject these beads into the cerebrospinal fluid, the CSF, and then they're taken up by the lymphatics and they drain to the lymph nodes. This is the normal route of any kind of waste material that's in the CSF. That's where they go. And then we do microscopy on these lymph nodes to look to see what percentage of those beads actually make it to this drainage area. So what Ashley found in our studies was even in a very mild head trauma situation, within the first day or hours, you have a very profound effect on lymphatic drainage, where the beads are not draining very much at all to the lymph node structures. You can actually see them in various regions of the brain. And it's not until almost 1 to 2 months post traumatic brain injury that the lymphatic function returns to normal. And this was somewhat of a surprise to us in the sense that we thought that it would return much sooner, especially in the case of a mild injury. And I'm not going to show it today, but at least the acute impairment that we're seeing in the lymphatic drainage function appears to be from the spike in intracranial pressure that happens post injury.
How do you know it's mild in a mouse?
Yeah, so you can use unconscious parameters, so writing time. So after you hit them, how quickly they return to cognitive function. You can also use pathology, so how much tissue loss. The other one is just a neuroscore. It's just like looking at their ability to like cross certain their activity, their ability to cross motor function across basically all kinds of different tests.
A bunch of scales that you use to classify.
Yeah, and then we have a bunch of like I guess maze related tests where you can evaluate their ability to learn, remember. That's the primary one, the combination of those. Does anybody else have any questions? Okay. So we become interested in wanting to understand why certain populations are so vulnerable to TBI. And two of the most, I guess, severe cases of TBI are those that occur with repetitive injuries to the head and also injuries in the elderly. And it's not really known why that is, but the thought is if you could figure out what's underlying the vulnerability in those populations, perhaps you could develop ways to eliminate that susceptibility in those individuals. So from Ashley's data showing that a mild injury, just one injury, can impair the lymphatics, we wondered if in the case of repetitive TBI, if these individuals already had an impairment in their lymphatic function before a second one came and it didn't repair itself, would that set that brain up to have more severe consequences? And in the case of the elderly, we know from data also that with aging, you have this precipitous decline in your lymphatic function. And this is thought to be one of the culprits in terms of aging being a major risk factor for neurodegenerative disease. So we were curious to know, in these two populations that have a very severe susceptibility to TBI, was a pre-existing brain lymphatic dysfunction what was setting them up to have worse consequences? So somebody that had a TBI before, if their lymphatics haven't been repaired, or in the age population, if they have an impairment, is that contributing to it? And this is just the aging data showing that aging is also associated with severe decline in lymphatic function. So this is comparing young mice, 2 months of age, to those that are 2 years of age. And this is similar to probably an 80-year-old person. The tracer here is in red, so you can see nice drainage in a young mouse and very poor drainage in an old mouse. And you can see it over here also. This is quantified, showing that there's impaired drainage with aging. So what Ashley wanted to address was, is it this pre-existing impairment in this drainage pathway that sets that brain up to more severe consequences? And we see these impairments, as I mentioned before, with aging and then also TBI. So the way that she did this, what we really wanted to hone in on was removing these lymphatic vessels to answer that precise question. So the way that we did that, we took a pharmacological approach where we injected in a compound called Visidyne. And this is used to treat macular degeneration, or back in the day, it used to be to treat macular degeneration. It goes in inert, but once you hit it with a laser of a certain frequency, you can get it to be bioactive, and it basically causes that vessel that it's in to rupture. So it's one way that you could potentially ablate the lymphatics in the brain without having to ablate the lymphatics elsewhere, which is an important thing that you want to do in terms of controlling limiting variables in terms of how you interpret your data. So what she did was she used Visidyne to ablate the lymphatics, basically injected this Visidyne compound into the lymphatics and then shined the laser onto various regions of the brain where we know the lymphatics are running to ablate them, let the mice rest for a week, and then came back, hit them with TBI, and then measured things like inflammation and behavior. So this is just showing you how it works. So this live one stain, this is basically detecting those vessels that are involved in the draining process. So that you can see the architecture of the lymphatics in the brain, above the brain. So this is just when you hit it with the laser alone. You can see that the lymphatics are in place. Visidyne alone without the laser, so it's just an inert compound. You have no effect on this lymphatic architecture. But when you inject Visidyne and then hit it with the laser, you can see you can get this really nice obliteration of the lymphatics, and this can actually last out to 2 months. So it's a pretty profound way to eliminate them. So what we did first was we looked 1 week post injury and just did an unbiased, comprehensive evaluation of the inflammatory status using a technique known as RNA sequencing. So you could basically evaluate the expression levels of the whole genome within the brain. And what we found was when you compared the mouse that just got a TBI versus a mouse that had that impairment in the lymphatics before the TBI due to that Visidyne drug compound, that when you look at things like inflammatory mediators, that many of these pathways are much, much more highly upregulating, suggesting that if you have a pre-existing impairment in this drainage pathway and then you get hit on the head, you're going to have a more severe inflammatory consequence from that injury. Just real quickly, I won't go too deep into this, but another way that you can evaluate inflammation is looking at something known as gliosis or astrogliosis. So when there's inflammation around, you tend to see upregulation of activation markers. So in this case, this is an astrocyte activation marker. So you can see that TBI or ablation alone induces activation of these glial cells, as you would expect. A hit to the head also upregulates activation of these, but you have an additive compound effect if that brain had an impaired lymphatic drainage pathway. And then one of the tests, and we've done multiple behavior tests and also learning, especially learning and memory tests. What we find is that those mice that had ablated lymphatics and then had a hit to the head have issues both learning and memory. This is just showing motor learning impairment. Okay, so that kind of provides some basis to explain why in repetitive TBI there is more severe consequences. So we're also interested in studying this as a therapeutic to be able to help those that are vulnerable in terms of in traumatic brain injury. And I should also mention that targeting this pathway is also being undertaken in translational studies for things like Alzheimer's disease and Parkinson's. So these concepts can also bridge over into other neurological diseases. There was actually a high impact paper from Akiko's lab, Akiko Iwasaka. She's at Yale. If you're on Twitter, she has like almost like 1 million followers. She's kind of the rock star of science Twitter these days. I'm not jealous of her, but she's fantastic. She's really down to earth, and she's just amazing. But anyway, in her lab, what she showed was if you boost the lymphatic function in a brain tumor, and then you come in with an immunotherapy, something that rejuvenates the immune response, so a PD-1 inhibitor, these you may have heard about some of these in terms of cancer treatment, that by boosting the lymphatic drainage and adding this, you are able to provide more of a beneficial effect in terms of eliminating brain tumors. So there's application of this biology to a lot of neurological issues. Okay, so what about the aged individual? Could we come in, try to recuperate or rejuvenate their lymphatic function, and then to make them more prone to a healthier, less detrimental disease course post TBI? So as I mentioned before, aging is associated with this massive decline in lymphatic drainage function, which you can see here. And so what Ashley asked is, if we were able to boost this lymphatic function in these aged mice, would this make them more protected against TBI? So the way that she did this was she used an adenovirus vector system. So this is basically a viral vector system that you could inject to overexpress a growth factor that can boost lymphatic function. So this is the compound that she was trying to deliver is something known as VEGF-C. It's a growth factor that causes there to be an expansion of angiogenesis of the lymphatics. And then with all good experiments, there was also a control to control for the adenovirus. And in this one, it just has a fluorescent protein known as GFP. So you can see here, this is an aged lymphatic vasculature. You can see that the architecture is declining. However, if you take an aged mouse and then you inject in this growth factor to boost the lymphatic drainage architecture, you can see an expansion of these live one vessels, both in terms of their diameter and also how much area of the meninges that they're taking up, showing that it can boost the expansion of the lymphatics in the aged individual. So looking at this as a potential way to limit the severity of TBI in the aged, we looked at this both in young and aged mice. And this is a phenomenon that's been reported extensively in the literature. Basically, if you take a young mouse and an old mouse, same physics, same area of the injury, same velocity of the impact, everything being the same, for whatever reason, the aged mouse is going to do much worse, both in terms of inflammation, pathology, and then also behavior. So this is just showing that here, comparing a young mouse to an aged mouse. And you can see that this aged mouse forms this glial scar, which is indicative of a more severe injury. However, if you were to give that aged mouse this VEGF-C compound to boost the lymphatic function before that injury, you are able to attenuate or ameliorate a lot of that neuroinflammation. And that's this shown here, where more severe consequences in the aged. But if you boost the lymphatic function in that aged mouse, you can eliminate some of that neuroinflammation. Okay, so more recently, we've also been interested in the phenomenon of how TBI can lead to an increased risk of Alzheimer's disease and other neurodegenerative diseases later in life. So it's pretty well known in the literature that suffering a brain injury can set your brain up to developing neurodegenerative disease later in life. So this is in the context of Alzheimer's disease, but you can also think about it in the context of CTE, which is a tauopathy that's seen in many athletes for post-repetitive head injury. So this is work that's now being conducted by a graduate student in the lab, Anna Arroyo Marco. I think she might be helping with some Spanish translation here, maybe in the coming weeks. But so Anna grew up in Spain. She came to the States to play tennis at Wyoming, and now she's a graduate student in the lab. And what she's been studying is she takes a mouse model of tauopathy. So basically, the mice express human tau, and then you hit them with TBI at a time point before they develop tauopathy. And her first question was, does the TBI accelerate tauopathy in those mice? And then the secondary, if you were to come and boost the lymphatic function with that VEGF-C that I explained earlier, would that be sufficient to eliminate the accelerated tauopathy that's associated with TBI? So she looked at behavior. She also did MRI to look at the ventricle size and hippocampal loss, and then also looked at pathology by imaging. And so this is MRI. This is looking at basically the hippocampus. So this is an area that's important for memory. So what happens is as the hippocampus deteriorates, these ventricles of the brain get larger to kind of fill that void. So this is a sham mouse at about I think it's 8 months of age. But comparing that PS19, that mouse that has tau, to a mouse that underwent TBI, you can see that there's quite substantial loss of the hippocampus, and that these ventricles get much larger, which is indicative of worse neuronal loss post TBI, consistent with what we see in individuals with CTE individuals. Interestingly, if she gives VEGF-C to boost the lymphatics one day after the TBI, she can reduce a lot of this hippocampal loss, and she can also reduce a lot of this ventricular swelling. And then this is just a control. So this is showing you here that TBI causes the ventricles to enlarge, which is indicative of neuronal loss, and that boosting the lymphatics one day after injury can rescue that. And you also see it with the hippocampus too. And going across the board, you can see that this rescues the pathology. So in the case of tau, what happens is tau gets hyperphosphorylated, and once it becomes hyperphosphorylated, it starts forming these tangles that cause neuronal death. And that's what you can see here. This is a stain for phospho-tau. So you get increased phospho-tau levels post-TBI, and then you can reduce this with lymphatic boosting. And then this is just another marker of tau. And it also has a profound impact on terms of the immune response in these mice too. So the conclusions from this part of the story are that TBI leads to impairments in lymphatic function. There's severe compromise early on, acutely, and this sustains itself, at least in the mice, out to 2 months of age, which was somewhat of a surprise. We've also found that pre-existing brain lymphatic dysfunction before TBI kind of sets that brain up for more severe consequences, both in terms of cognitive function, inflammation, and pathology. And then our work is also showing that boosting lymphatic function can provide some protection against TBI severity and aging, and also limit tauopathy that's associated with TBI. So I'm gonna kind of shift gears for maybe the last 10 minutes or so. But if there's any questions on this part of the talk, maybe it's a good time to take a quick break to answer those or not.
Has this been shown in humans?
Yep.
So the TBI leading to impairment in brain lymphatic drainage?
That is still in progress.
Okay.
So I guess one thing to remember is, so the lymphatics were first discovered 10 years ago, and it took from that time point to publishing their existence in humans, it took about 5 years.
Yeah, that makes sense. I just can't believe it's only been 10 years. That's
Yeah.
so crazy.
Yeah, so it's taken a little while. They've done these tests in Parkinson's disease patients where they've looked at lymphatic compromise, and it correlates with Parkinson's severity. They're starting to do more and more of this. It's just having the technology to be able to evaluate it.
Right.
So it's come a long way, but there's still so much that needs to be done to kind of evaluate it more.
Mm-hmm.
Yeah, it would be interesting. Like I've always kind of, I mean, this is a bad way to look at it, but obviously when you turn a certain age, you go in and you start getting colonoscopy. You start, there's certain things that you do to see how things are working.
Right.
It would be the ultimate would be like going in and learning how the age of your brain based upon your lymphatics, and adjusting that if that's a situation that could set you up for more severe consequences.
Mm-hmm.
And in the case of TBI, for a lot of these, especially young athletes and even professional athletes, we really, the objectives to sending them back out to play are very subjective. A lot of people actually cheat on their pre-tests so they can get back on the field. So basically before the season, you have to answer a bunch of questions and do motor tests, and people usually, or some individuals will actually perform worse on that just in case they do have a TBI, so their baseline's lower. But for this, it would help to address how severe that injury was, and then how long it took them to return to a place where it was safe to go back out into, I guess, an impact sport or a situation like that.
I have another question.
Yeah.
So that the laser technique that you mentioned to ablate the lymphatics.
Yeah.
It sounds a lot like sonodynamic therapy in some ways, which is where we take a non-toxic dye essentially, and then use focused ultrasound to activate it to cause tumor cell death. What is the mechanism behind that? Is it that it activates an apoptotic pathway?
Yeah.
And then they're able to grow back though?
Yeah, so it induced a reactive oxygen species within the lymphatics.
That's exactly what's on.
Yeah, but the interesting thing with the brain lymphatics is unlike other lymphatic vessels elsewhere, the brain lymphatics are very sensitive. They require tonic VEGF-C to maintain themselves.
Do they have high turnover?
Yeah.
Yeah, okay. That makes sense.
So another way that people are doing this now is they'll either put in like antibodies to VEGF-C in there, or they'll put in viral vectors. And it basically sops up all the VEGF-C, and the lymphatic vessels basically deteriorate and are not there. If you were to do that, say if you did that in your skin, your lymph nodes that are collecting there, you really wouldn't have any problems because it's more stable. So for whatever reason, there's the the physiology of the lymphatics above the braid are different than the lymphatics elsewhere.
Hi, John. Sorry, go
Okay,
ahead.
go ahead.
Hi, sorry. This is Susie LeBlanc. I'm on Zoom. Great presentation so far. Is there a linear relationship between how much the lymphatic system can drain, like according to the size of the traumatic brain injury? Is there a limit to how much it can get rid of toxins or open up?
So if I understand the question correctly, we do see a linear, I guess it correlates with severity. So if we were to increase the severity, basically the compromise would be more severe and the impairment would last even longer than that 2-month time, whereas we dosed it down to make it even less severe, so really, really mild. We'd see an impairment acutely, but it would return sooner. So it does track with severity quite well.
Okay. So I'm just thinking down the line with like the amount of amyloid burden, when we want to treat amyloid either earlier or later, would it be better to treat earlier when there's less disease, or can you also treat later when there's more disease and the lymphatics will pick up the extra disease burden?
Yeah. Yeah, I guess part of the problems with later in disease, I mean, there's obviously a lot of issues to think about. One is like, have you already passed the point of no return where there's been so much loss of the neurons? And then when it comes to the amyloid beta, as the longer it's there, the more dense and the larger the plaques become. So that, I mean, that's never going to fit through the lymphatic vessel. It's more like the soluble, monomers and also oligomers. So you might not be able to touch much of the big plaques in lymphatic boosting. It's going to mostly be the smaller oligomerics, and then also removing other things that are associated with the damage around the neurons that could also kind of trigger the immune system to kind of perpetuate the disease.
So is there a size kind of limitation of what can
There
cross into the
is.
lymphatics?
There is. I mean, there is basically they're only so large, yeah, so there is that issue.
Okay, thank you so much.
All right. The interest of time, I'm going to move on, but I'm going to be very disappointed and depressed if one of the residents or the younger folk don't ask a question. I'm just going to put that out there. But anyway, okay. So now, just very, very quickly, I wanted to talk a little bit about. So that's more at the systems level, the higher level of trying to increase the drainage pipes. How could we actually modulate the cells that are kind of right there in the battleground, in terms of that are engulfing and also containing these neurotoxic materials? Some more cellular-based therapeutics. So our lab and others has gotten really interested in this idea of microglia playing an important role in AD. And it's really only been in the last 15 to 20 years that we've come to realize that AD is a disease associated with microglial compromise. And a lot of this has come from genetic studies where we've looked at genetic factors that are linked to late onset Alzheimer's disease. And many of these, and this is an old graph, it needs to be updated, but many of the genetic risk factors are associated with the immune response. And this was quite a surprise to the field when it first came out. At the time, it was thought that Alzheimer's was just a disease of poorly behaving neurons and not so much the immune system. And in addition, so this graph, when it does get updated, many of the genes that aren't actually characterized as quote unquote immune genes are almost exclusively expressed in microglia. So two of the other genes that we're working on, I'm not going to talk about them today, are RIN3 and Cas4, which are here, which don't come up as immune genes. But if you actually look in the brains, you pretty much only see them being expressed by microglia and not other brain cells. So the field has really changed its focus in the last couple of years to looking more at these quote unquote glue cells and their abilities in terms of disease. And you also see this in the patients too, where you just have this massive upregulation of these microglial cell types in very severe forms of neurodegenerative disease. So one of the molecules that we recently turned our attention to was a molecule called IMPP5D. So it is a genetic risk factor for late onset AD. This is it over here. So this is, and I'll get into this later, but this is a project that we're collaborating with Rich Price on, who many of you may know from his seminal work in the field, Focus Ultrasound. But we're actually co-mentoring a student together, Josh Samuels, and what he's doing is with me, he is kind of establishing what the important players are, and then with Rich, what he's doing is he's developing genetic-based systems to target those and using focus ultrasound to actually get them into the brain. And this was actually the idea of the graduate student who really enjoyed both our labs and wanted to do something a little bit more translational. So to study this, what Josh did was he knocked out this gene, IMPP5D, which is a phosphatase in microglia. And I'm not going to get into the actual genetics of it. I can talk about that later if you're interested, but it's not important, I don't think, for this talk. But when you get rid of it and you put it on this 5XFAD background, so what these 5XFAD mice are is they have multiple knock-in mutations where they produce really high levels of amyloid beta. So we've basically knocked in multiple mutations that are seen in patients that have Alzheimer's disease and that produce abnormal amounts of amyloid beta. So this is one of the tools that a lot of us in the field are using to study Alzheimer's disease. So what he found was that the microglia basically do a better job of swarming these amyloid beta plaques. So the plaques here are in thio-S, so the pink areas are the plaques, and then IPA1 is a marker of microglia. And so basically they converge onto these plaques to help eat them up, but also to almost form this like almost a protective cocoon around them. Because you have to remember this amyloid beta is neurotoxic. So you want to limit the surface interaction that it has with neurons, which would be susceptible to it. So we see an expansion of the microglia. They're larger, and then there's more microglia per plaque. And this knocking out this gene has a beneficial effect in terms of limited neuropathology. So this is an APP puncta. This basically marks dystrophic or unhealthy neurons. And so you can see here in the wild type, there's lots of unhealthy dystrophic neurons surrounding the plaque. But in the mice that lack this molecule IMPP5D in their microglia, on the 5XFAD background, you're able to ameliorate a lot of this neuropathology. So what Josh is doing now is trying to figure out how this is happening. Microglia can protect the brain in various different mechanisms. One is recruitment. One is phagocytosis. Also, you can have expansion, so an increase in numbers, and that can do a beneficial job. There's actually quite a bit of literature on this topic in terms of focus ultrasound in the literature. We're basically using focus ultrasound to quote unquote activate these cell types will help them phagocytose and engage with the plaque more beneficially. And there's been multiple publications on that. But interestingly, Josh, to our surprise initially, these IPP5 deficient microglia don't do a better job of clearing plaque. So that you can see more plaque. This is the red stain, the D54D2. Also by ELISA, there's more amyloid beta. But what's happening, or at least what we think is happening, is that the microglia are just really, really good at forming these almost like granulomas. So the immune system does this in infection too. So in some infections, what the immune system will kind of build this fortress around it to kind of hold that dangerous pathogen in. So we think what's happening with this deletion is the immune system is doing a very similar thing. forming this almost complete coverage around this neurotoxic material so that none of it could potentially interact with the neurons. And then this also leads to those microglia being able to better compact that amyloid beta. So Josh, along with Rich, we've also been trying to tease apart other molecules within this pathway. And the thought is, like, can we identify breaks that are slowing down the microglia so we could take them off so that they could have more of a beneficial and neuroprotective role in protecting the brain against neurotoxic material. So this is another molecule that Josh identified based upon some screening. And you can see here, this is looking at thioester, this is amyloid beta. And then if you look at deletion of PTPN6 and actually encodes for the protein's name is SHIP1, you can see that by deleting it, it does a fantastic job of limiting the amyloid beta deposition. And like we saw before with IMPP5D, we see that there's more microglia that swarm onto these plaques. So the purple here is a stain for microglia. Amyloid beta is here stained in blue with thios. And then, as you would expect, with very little amyloid beta or neurotoxic material in the brain, there's very little neuronal damage. Oh, and I didn't show it, but how this works is basically promoting improved phagocytosis of the amyloid beta. So, so the question is, well, why not just give a drug that inhibits these two molecules? And the issue is, and this is going to be a major thing that we're up against in the brain, especially targeting the immune system, is if you were to hit these molecules globally, you would have a rampant immune response. So you would have an autoimmune and autoinflammatory disease. So what we're trying to do is, along using FUS, kind of leveraging that to target these compounds so they only become active in the brain so we can limit this collateral damage in other organs. So that's kind of what we're doing alongside Rich Price. Yeah, so this is Josh. He's a third-year graduate student. He's doing most of the AD work that I showed today. Anna's here. She did the tau work, and then Ashley's here. She did all the initial TBI work. And then this is our whole lab. I have most, Neil has his certain attire and certain tendencies, and with every boss, we all have our nuances that we like to joke around about. I have mine too, and one day the lab all dressed up like me. And this is a picture from that. I have a very some very normal, well, they're not normal. Jessica can tell you that, but that's just everybody from our lab. But anyway, like I said, the interns, you got to ask a question. I'll get Neil in here.
What's the function of IMPP5D in wild type mice?
So it's a phosphatase. So basically proteins become active when you phosphorylate them with kinases. So it's basically going in and removing those phosphorylation sites. So it's thought to be a major molecular break to shut down immune responses. So that's kind of, and it can function in macrophages, microglia, like we're doing, but it can also function in neutrophils and T cells, and so that's kind of how it works. Yeah.
Do we treat all of the neurodegenerative diseases as like TBIs, or are there other ways to approach it? And by treating it as a TBI in the way that you're experimenting on mice, are you possibly missing the mark on how that works with lymphatics?
Yeah, so I think, so it's a good question. It's kind of a larger question. I think you could, so looking at some of the early data from the experimental models, aging, Alzheimer's disease, ALS, Parkinson's, boosting lymphatic function in all those cases has a beneficial role. And then, there are certain common themes that can be targeted across the board, and that seems to be one of them. In terms of TBI, that's only a small percentage of individuals that have a neurodegenerative disease. That's only the culprit in a small fraction of them. I shouldn't say small fraction, but there's different slivers. So that's an important point in the sense that no patient is identical in terms of how you want to treat them, because how they got there is likely going to be different. They could be an individual that have a loss of function mutation and also had a TBI or didn't have a TBI. Or, there's other environmental risk factors that can also come into play. Things like exposure to pesticides, radiation, obesity, and lack of exercise also kind of factor into. So, one thing that people are thinking about, and they're doing this a little bit better in cancer right now, is precision medicine, understanding what the mutation is. And then, also the stage of the tumor, and then developing drugs around that. Unfortunately, we're kind of in a bad position with a lot of neurodegenerative diseases because we really don't have anything to offer these patients that target the disease-modifying culprits. We have things that can help with their symptoms, but nothing that's going to slow or reverse disease. So we don't even have anything to diversify in terms of our arsenal that we treat them with. So that's why some of the advancements that we're seeing with focused ultrasound and those antibodies provides a little bit of hope, but we're going to need other things, especially considering that when we do start treating patients, it's usually at a time where they've already lost so many neurons. We need to develop ways to identify them earlier. So a long answer to your tough question.
That's good.
I'm not an
Yeah,
intern,
yeah,
I have
yeah,
a question.
yeah.
So there was an article in the New York Times recently, sort of hit close to home for me because my mother's 96 and she's just super sharp,
Yeah.
better memory than anybody in the family, right? So are there any of these mice that show up are like super agers?
Yeah, so I mean, some of these like IMPP5D, PTBN6, like they're super agers in terms of the amyloid beta pathology. That's this interesting that you bring that up. There's been more of a push in the neurodegenerative field to study those individuals as opposed to studying the sick individuals. Like,
Yeah.
what is it about them that makes them resilient? Because and it seems to be exercise.
Okay, not a factor.
Yeah.
Dang it.
Sleep's a big one.
Jimmy Dean's.
Sleep.
She still drinks.
Sleep is a big one.
Okay.
So basically, this lymphatic and the lymphatic pathway that kind of pumps into lymphatics, that's most active when you sleep.
Okay.
So if you have fragmented sleep, you're basically shutting down, or you're reducing the capacity of the system. And then there's so many different things, but there are things that we know that can help the aging brain, and definitely sleep and exercise are two of the best. But yeah. Yep.
You mentioned some like external factors, like risk factors, whether it was, you said pesticides, radiation, lifestyle, things like that.
Yeah.
Is that considered to be like plaque buildup from that? Is that considered to be like amyloid buildup? Is that considered to be irreversible, or if it's at like a certain age while the lymphatic system is still healthy, or is that able to be sort of like reversed to some degree?
Yeah, I mean, the sooner you catch it, the better you are in terms of addressing it. Obviously we're all exposed to these things in life, some more than others, obviously. And then, some individuals, there's other variables in terms of they might have loss of function, SNP mutations, and a gene that we're studying and not know it. And so they're even, that takes their threshold to a different level. But if you were able to catch most of these early time points, you would, because a lot of them are chronic progressive, right? In the sense that they keep on building onto themselves until they cross that threshold. And then it's harder to address. So if you can catch the early seeding stages by boosting lymphatics, you have much better shot to kind of catching it before it becomes a big problem.
Any other questions in the room? Anyone online have any questions? We can't necessarily see you, But if you want to ask John a question, we can hear you.
Or you could turn on your video.
You know how I love to ask questions.
Yes.
I'm sorry if I missed it. I had to step out of the room just for a moment, but I was wondering if John was familiar with Hong Chen's work and if they had worked together at all on the lymphatic drainage studies.
She's at WashU.
Oh, she's at WashU? I work with Yoni Kipnis at WashU.
She's doing a lot of liquid biopsy.
Oh, it's liquid
research, but
biopsies,
I
yeah.
don't know. Yeah, I was actually thinking about
She
it.
does lymphatic drainage also.
Yeah.
Oh, okay.
Yeah,
Yeah.
so
Yeah,
Oh,
I don't know. I don't know her specifically. I'm headed there in October for a conference they're having. Maybe I'll meet her there. I'm sure she's interacting with Yoni as long as she can put up with him. Yeah.
yeah, I can send you some of her work to see if you have anything in common. She's really interesting.
Yeah. Hi, John, this is Susie again.
Okay, hi.
Along the same, again, thank you so much. This is so fascinating. And in the clinical work that we're doing with focused ultrasound and Alzheimer's disease to increase microglia and probably inadvertently, possibly increasing the lymphatic drainage, on the PET scans that we've been able to do so far in clinical studies, there has been shown the ability to decrease the amyloid in the areas that are targeted with blood-brain barrier opening. Again,
Yeah.
whether it's a combination of the microglia infiltration and or improved lymphatic drainage. But I'm very curious now about, when we want to, like you said, like treat earlier, either one day, maybe we'll be able to catch the disease before they're clinically symptomatic, but we have enough liquid biopsy markers to know that they're on their way.
Sure.
But is there any way to help quantify in preclinical models based on the size of the amyloid and tau depositions per cubic submillimeter, I guess, in animals, how much we can really decrease the amyloid and tau in those regions and subsequently improve the neuronal function like this? And I'm not a preclinical researcher, so I apologize if I'm not phrasing this correctly, about how much we can preserve or restore neuronal function around the size of the plaque per plaque size density. Do you know what I'm saying?
Yeah, yeah. Yeah, I mean, it kind of brings up a larger issue in the field that, most people kind of avoid in the sense of like, there's a lot of people walking around right now that have a ton of amyloid beta in their brains and are completely fine. So I think there's still an inadequate understanding of like what the actual most toxic form of amyloid beta is. It's thought that more of the oligomeric species can actually do more of the damage. And then the other question becomes is, the amyloid beta is actually not homogeneous. There's quite a bit of junk in there too. So there's a lot of RNA and DNA, and then there's a lot of heavy metals. So everything from iron to copper. And there are a lot of individuals that are starting to trying to better identify like what components of those plaques are more toxic than others, because they're the ones that you really want to key in on. And I think once we get a better handle on that, I think it would be easier to kind of assess, how well we can do with handling it.
Okay, thank you.
Anybody else? Any questions?
I might have
Yeah.
one. Yeah. If I think you like hit the mouse, but how do you keep the consistency each time?
Yeah.
How do you hit?
Yeah. So there's probably like a couple hundred different traumatic brain injury models. Everything from there's fluid percussion, so you could actually push fluid through the brain to cause this like shear force. Back in the day, they used to use like weight drops. There's methods where basically you do rotation injury to kind of whiplash, but how we do it is we have an impactor where you can control the velocity of how it hits, how deep it goes, and then you can change the head's diameter and also the composition. So like a rubber versus like a steel. And then you put it on the stereotactic frame, and then you position the head in a kind of a rig. So you can kind of use coordinates of the brain to know, the hope is you're going to hit the same area every time. And that's kind of how it works. But it's there's companies out there that generate these impactors for these types of things.
One more question.
Yeah.
For the BEG study, there was like, I'm not sure if it was significant, but there was little difference between sex.
Yeah.
Is that, I mean, when I thought it might be because like the size of the organ differs from male to female, and it might cause like bigger organ, maybe slightly higher function, therefore. But was there, what did you see over that? Or it was, I don't know.
Yeah. So you know, the role of sex in disease is something that has gained a lot more attention since I've been doing research, and it's for good reason, because we see incredible sex differences in almost every disease where the immune system is involved. You know, you look at things like lupus, which is so female dominated, tauopathy is male dominated, and then we see that at least in our amyloidosis mouse model, we see that females get earlier accelerated disease, and it's actually aggravated compared to males. Whereas that tau model that we're using is the males that get it first, and it's really bad. So understanding why that is I think really important. And I guess it goes back to your question about how we treat. And some of it has to do with you know, sex chromosomes and what's on there. Some of them have more or less of certain immune genes. But yeah, it's something that needs to always be considered. We've never done a longitudinal assay with our mice, and nobody's done it in humans yet, to see if there are differences in terms of lymphatic compromise based on sex. We've only done like 2 snapshots, early and late, but it could be that, you know, one or the other deteriorates a lot faster than the other.
Okay. Well, I just want to say thank you again, Dr. Lukens. That was a fantastic talk, and I definitely learned so much and have so many other questions that I'd like to ask you. This will conclude the speaker seminar series for today.